invited talk. Hello, everyone, and welcome to the Kite Research Institute invited talk given to us today by Professor Qingyu Zhao of Stanford University. It's my great pleasure to introduce our speaker, who's a faculty member in the Department of Psychiatry and Behavioral Sciences at Stanford University. He received his PhD in computer science in 2017 from the University of North Carolina at Chapel Hill. Dr. Zhao's research has been focusing on identifying biomedical phenotypes associated with neuropsychiatric disorders by statistical and machine learning-based computational analysis of neuroimaging and neuropsychological data. Dr. Zhao received a Pathway to Independence Award from the National Institute of Alcohol Abuse and Alcoholism in 2021. Please join me in welcoming Dr. Zhao in a virtual round of applause. If you have any questions, please type your questions in the Q&A box in Zoom, and Dr. Zhao will answer them at the end of the talk. Thank you, and welcome, Dr. Zhao.
Thank you for the introduction. Let me first share my screen. So I believe this will work. Okay, so first of all, thank you for having me here. I'm Qingyu, and I'm instructor in the Department of Psychiatry and Behavioral Sciences at Stanford University. Today, I'll be mainly talking about some of the computational approaches, mainly the deep learning-based ones that we have developed over the years in our lab to support longitudinal neuroimaging studies. Okay, so before actually going into that topic, I'm just gonna very briefly introduce our lab. So for some of you who may not know, I'm actually affiliated with the Computational Neuroimage Science Laboratory, where I mainly collaborate with Dr. Kilian Pohl and Dr. Asan Madali on conducting some of the computational neuroscience research works. And in our lab, we aim to use unbiased and interpretable machine learning approaches to analyze biological, neuroimaging, and also neuropsychological data in order to improve understanding of normal neurodevelopment and also some of the developmental courses of some of the neuropsychological diseases and their interactions with daily behaviors. And in order to achieve that goal, it is very important to be able to conduct longitudinal studies to track individuals over time. So the first question is, what is a longitudinal study and how it is different from its counterparts, namely a cross-sectional study? Well, in cross-sectional studies, one generally recruits a large cohort of participants and then collects data from them. This data collection procedure usually happens just once. So basically the collected data is like a snapshot image at a particular time frame. And usually because of the high heterogeneity of this population, cross-sectional studies are often used to characterize group differences or to find association between disease outcomes and certain factors of interest. And longitudinal studies, on the other side, are able to track those individuals for a more prolonged period of time by repeatedly taking measurements from them. So basically, longitudinal studies are very useful to analyze time-sensitive or to answer those development-related questions, because apparently we can analyze those temporal trajectories associated with the individuals. Okay. And because of these particular strengths that I just talked about, you can actually see a surge of new longitudinal neuroimaging studies being initiated after year 2000. And we know that the longitudinal design is really powerful, but actually conducting a longitudinal study is much more challenging compared to conducting a cross-sectional study. And the challenge mainly comes from two sides. And the first one is basically the difficulty of data collection. And typically there are just fewer people that can commit years of participants in a longitudinal study. So you can see that the sample size of a typical longitudinal study is relatively small. And those participants who initially want to, attend or participate will somehow drop off in the middle. So the severity of missing data will drastically increase as the study goes on. And there are other factors, difficulties in data collection, but I guess I'll stop here because that's not the focus of this talk. And the other challenge I want to mention, which is the focus of this talk, is that we need to essentially adopt more sophisticated computational approaches to analyze those longitudinal data, because the data itself is inherently more complicated. And what I mean by that is that if you look at this a typical analysis in a cross-sectional study, if you look at one measurement or one feature in that study, you can see that the variance basically all comes from the inter-subject difference in the underlying population. But now if you look at this right figure, or the spaghetti plot recording the longitudinal trajectories of those participants, you notice that the variance in that measurement not only comes from the heterogeneity of the population, but also comes from the changes within each single trajectory of each individual. In other words, basically you have this mixture structure of data variance in longitudinal studies that our analytical method has to properly recognize. And to correctly model such mixture of variance, we have developed a series of tools to analyze longitudinal neuroimaging data for different applications. And first, we know that neuroimaging data basically needs to go through a series of sophisticated pre-processing pipelines in order to get some reliable measurements before those measurements can actually go into the actual analysis stage. And it turns out having this pre-processing pipeline specifically designed for longitudinal data is very critical for improving the statistical power of the study. So I'll briefly introduce one work along this line of thinking. And once you have those clean data to start analysis with, we can then choose different analytical models, such as usually the case is choose a random mixed effect models. But today, of course, I think I'm gonna talk about the deep learning ones, especially on how to use the repeated measure design of a longitudinal study to learn better image representations of MRI data. Okay, so before actually going to the deep learning works, I do want to very briefly talk about one work that we did on producing longitudinally consistent data, because I feel like this is going to be very important for all the follow-up deep learning analysis to be able to properly work. So to motivate this, imagine you have measurements that do not show any consistency between visits of subject. For example, in the left figure, then it's essentially very hard to determine any meaningful developmental pattern on the group level. On the other hand, if you have less noisy individual trajectories, it would be much easier to do the group level inference. For example, on the right figure, it's clear that you have a quadratic developmental pattern. And in fact, I generated these two synthetic curves using the same parametric model. And the only difference is the noise level. So you can see that the noise level can really make a huge difference when you try to interpret these data. So this is our motivation, that we want to consider dependency between the repeated measures within each subject trajectory. So we have made a lot of efforts in designing such longitudinal pipelines. And one of them that I want to very briefly mention here is how to generate longitudinally consistent functional networks from resting state functional MRI data. So for those of you who are not quite familiar with functional imaging, it's basically a 4D MRI imaging modality that describes the temporal brain activity. So that is the so-called boat signal recording the oxygen level in each brain voxel over time. And this resting state functional MRI refers to the recording of spontaneous brain activity when there is no explicit task being performed during the scan time. And a major usage of a resting state functional MRI is to characterize the functional organization of the brain. So, or specifically to find these intrinsic brain networks that basically describe which brain regions are co-activated together and which brain regions are deactivated together. And these networks, with these networks, we can basically investigate how the brain functional profile develops over age, or whether there's abnormality associated with certain neurological diseases. And as you may know, there are actually a lot of ways to quantify these intrinsic brain networks. And one of the most popular approaches for estimating such functional networks is based on independent component analysis. And the idea is that the resting state bold time series of all voxels, which can be written in this matrix form, can be decomposed to the activation of a few functional networks. And here, each component in this matrix corresponds to one brain network. Basically, it encodes a whole brain spatial map describing which regions are co-activating together. And the corresponding component in the left matrix records the temporal activity pattern associated with that specific network. So essentially, the upshot is that with one MRI, resting state functional MRI, we will be able to derive a few functional networks with this ICA approach. Okay, and let's say we have a longitudinal study to examine how each functional network develops over time. And the traditional approach would basically perform ICA to each individual visit of a subject separately. For example, you can derive a specific network for the first visit by applying an ICA, and then you apply this ICA again to the second visit to derive another network, and then for the third visit, you do this all over again. However, due to the substantial noise in functional neuroimaging, it is very likely that the resulting estimates exhibit huge variation, sometimes even biologically implausible. For example, if you look at a specific voxel, its connectivity strengths may be really noisy, exhibiting a really large variation in this trajectory. And so if you now put together all the individual trajectories, it's almost impossible to do a group level inference because you just have a lot of uncertainty in your data. And the trick we realize here is that, well, in the end, we're going to use a parametric model to perform this group level inference, to figure out what is the group level trajectory. For example, you would assume that you want to now find a linear or quadratic developmental effects on the group level. Well, if you assume it is a linear on the group level, you may as well apply the same assumption in the first place to derive subject-specific trajectories under the same constraint. So the idea is that now we want to jointly perform this ICA for all the visits together instead of doing it one by one. And this allows us to put additional constraint in the estimates. For example, you can encourage the connectivity strength at each voxel to follow a linear trajectory across visits. And then basically we can put this regularization together with the original regular objective function of ICA. And by doing so, essentially we limit the search space because when you try to estimate networks from one visit, you're leveraging information from other visits so that the joint estimate can be more robust against noise. And in the end, if you apply this approach to derive longitudinally consistent networks for each individual, and you put them all together, you have a much easier time during the group level inference because the noise level is significantly reduced. And we apply this framework to examine the developmental effects of functional networks over the adolescent period using the resting state data from the Encana study. Just so you know, the Encana is basically one of the largest multi-site longitudinal neuroimaging study in the United States for studying the adolescent brain. I guess I won't talk about details about the results here, but the upshot is here that we showed that our approach is more sensitive than a lot of other approaches in detecting subtle developmental changes in these functional networks. Again, I'll stop here for this work, but because I want to spend a little bit more time on the deep learning work that will come next. So let's say we use those longitudinal consistent measurements or MRI data, you're ready to apply this deep learning analysis for different applications, because we all know that deep learning is attracting more and more attention in neuroimaging studies nowadays due to its flexibility and sensitivity. So basically, in every small application where deep learning is claimed to be applicable, there are people showing deep learning approaches can win against conventional approaches, which is astounding. And as a result, there emerged a lot of ways that deep learning can be embedded into a neuroimaging study. And a very common workflow is something like this. So assuming you have a trained deep learning model and you input a raw MRI data, a raw MRI image into this model, which can automatically extract features from the MRI data. Here I call the extracted features the latent representation of MRI. And depending on the final application, this representation can be used to perform downstream tasks, for example, tissue segmentation, tumor segmentation, or classification of diagnosis labels. And we do want to note that the most difficult part of this workflow is to be able to train this deep learning model in the first place. And this training often requires a ton of labeled ground truth data. And what I mean by that is, well, if you want to do this segmentation, you basically have to have a lot of existing MRI data whose correct segmentations are known beforehand. Or if you want to do this classification of diagnosis label, you then need to have a large cohort of participants where their diagnosis labels can be made before training model. And if you don't have enough label data, I guess it's going to be really challenging to learn credible latent representations of your MRI data. And so this challenge stimulates the advancement of one research branch named self-supervised learning. So unlike in traditional machine learning approaches where you just directly train the model on label data, in self-supervised learning, you first train your model on this so-called pretext task. So basically, it's a task not of your primary interest, but it's a task that you can easily get some supervision signal for. For example, you can train a deep learning model to predict whether two patches of MRI data come from the same subject or not. I'm just giving a random example here, but you see that this pretext training does not rely on any external labels, because all this supervision signal comes from the data themselves, so that you can basically use all the available data at hand. And once you train this pretext model task, if you will, you already got some semi-ready latent representations underlying the MRI data, and then you can further fine-tune your model on the downstream task, which is your primary task of interest, and this training will be using the actual labels of your actual task. And recently, the idea of self-supervised learning has been used for many neuroimage analysis for different applications, but most of those applications are cross-sectional studies. So in this talk, I'm gonna try to address the fundamental question that how do we learn image representations from longitudinal MRI data, and how does longitudinal data provide unique self-supervision signal that cross-sectional data cannot provide. So basically the question boils down to, well, what does longitudinal data have that cross-sectional data does not? And we realize that the essence of the repeated measure design in a longitudinal study is to essentially better disentangle time-related developmental changes from other static factors in the study, and that's the essence, of the longitudinal study. And what I mean by that is that if you look at a cross-sectional study, and if you look at the difference between any two samples in the data set, and that difference could come from a lot of different sources, for example, sex, age, race, etc. But now if you look at two samples or two visits of the same subject in a longitudinal study, their difference is only linked to time-related dynamic factors, and all the other static factors such as sex and race remain the same during the study period by construction. And that means if we focus on analyzing the within subject difference over time, we have a better chance to disentangle time-related factors from those time-independent factors in the latent space. So based on this notion, we developed a framework called longitudinal self-supervised learning. And I did this work together with Zach Liu, who is now a PhD student at Washington University. So the idea here is that longitudinal data is primarily used for quantifying developmental changes. So here we make a simplifying assumption that the only factor that induces changes across visits of the same subject is the factor of brain age. And here, when I say brain age, I do not mean chronological age, but brain age is actually a marker quantifying the health condition of the brain. So basically, people with the same chronological age can have different brain ages. And for example, people with neurodegenerative diseases could have higher brain age than healthy controls, despite that they can have the same chronological age. So with this assumption, what we can do here is that we can build this autoencoder system to learn the low-dimensional representations of MRI data. I assume some of you may have worked with autoencoders, but basically they're just an encoder neural network that can reduce, that can encode the MRI data to a latent representation and then use a decoder network to reconstruct the exact input. And now the idea is that in that latent space, we can also learn a 1D direction linked to the factor of brain age. And how we learn this direction, or how we disentangle this direction from the latent space, is that we take a pair of images from the same subject. So the pair of images is from two visits of the same subject, and we encourage their progression direction to be parallel against this brain age direction, which I call tau here in this work. Again, because this parallelism is motivated by the assumption that the only factor that changes over time is this brain age for each individual. In other words, the difference between Z1 and Z2, two visits of the same subject, should fully align with this global direction tau. And basically, we can apply this regularization for all pairs of within subject MRIs, so that we can jointly estimate all the latent representations of MRIs and also jointly estimate this global latent direction tau. And in the end, you have a more regularized, more structured latent space where one direction is explicitly disentangled with the factor of brain age. And just to give a very high-level idea of the training objective function, is just that in addition to this regular autoencoder loss, we added these cosine losses to encourage the parallelism between the progression direction of a visit pair of an individual and the global direction tau. And in this training, I want to note that it's fully supervised, because this training task can be regarded as a pre-test task for any application, because we do not need any external labels to train this model. We do not even need the actual chronological age of the subjects to be able to train this model. And what we need is only the information that which visits belong to which subjects, and this supervision signal comes from the data itself, independent of the task. I mean the final clinical application. And moreover, this training uses pairs of input, so it typically do not have the issue of not having enough training samples, and this turns out to be a big strength of the method too. And lastly, I would assume that the specific architecture and the implementation is not of too much interest here, because we can find the code on GitHub online. Okay, so we validated this model, LSSL, longitudinal self-supervised learning, on 2 longitudinal neuroimaging datasets, and the first one being ADNI. And for those of you who are not fully aware, basically ADNI is one of the largest neuroimaging dataset for studying the effect of Alzheimer's disease and also studying general cognitive, the effect of general cognitive impairment on the human brain. And essentially each subject was repeatedly scanned over time, over several years of the study period. So basically it provides a really good setup for our analysis. And what we did here is that we trained our model on all the longitudinal MRIs, including all the subjects, including the normal controls and participants with MCI, that is mild cognitive impairment, and also patients with Alzheimer's disease, because the LSSL is a self-supervised method. It does not require any label information, so you can use all the data at hand. And there's no training test here because, again, the model is self-supervised and does not require any label. And in the end, we can get, after training the model, we can get all the latent representations of those MRIs. And for each MRI, we can project its latent representation onto this direction tau, which is also estimated by the framework. And this projected value, which is the scalar, is then the estimated brain age associated with that MRI. In other words, if this projection point is more towards this direction, it means the MRI has a higher brain age, otherwise it has a little lower brain age. And we can then basically plot all the estimated brain age, and all the estimates, all the brain age estimates, and plot those estimates with respect to their real chronological age. And we can do this plot first for the healthy controls, and we can see that there's a clear correlation between the estimated brain age and the true chronological age for the healthy control cohort. And this is to be expected because we know brain age generally aligns with chronological age for healthy controls. But you notice that there's still substantial heterogeneity for any given chronological age, in the sense that people of the same chronological age can have slightly different health conditions in their brain. And interestingly, if now we plot the estimated brain age of the AD patients, the patients with Alzheimer's disease, on top of the healthy trajectory, we can see that their brain age, which is indicated by the red dots, is generally higher than their chronological age, indicating the accelerated aging effect of the disease. And so in other words, the Alzheimer's disease would make a brain looks much older than what it should be, because the disease induces neural loss, which causes accelerated cognitive impairment. And moreover, it's quite clear that this gap between brain age and chronological age is more pronounced in the younger cohort. And this is also where the accelerated aging effect is the most severe, in the younger cohort. On the other hand, if you are already an oldest old and then gets Alzheimer's, the disease effect is more negligible, because you're already at a late stage of cognitive decline, and the phenomenon is, the young people specific disease effect is very frequently reported in AD literature. So it's quite encouraging that we can get this result fully by this self-supervised method. Remember, the training does not require any label information of the data, yet it's still able to separate against the normal controls and people with cognitive impairment. And also, just to give an intuitive idea how accurate our estimated brain age is, essentially we select a few people with the same chronological age, which is 80 years old. And interestingly, we can see that although they're the same old, their brain looks quite different, right, of different health conditions. And now for each image, now we show its estimated brain age on top, and we can clearly see that our estimates align with the typical aging pattern of the brain observed in the literature, which includes the enlargement of ventricle and thinning of the cortical gray matter, essentially the atrophy phenomenon. And on the other hand, if we pick those people with the same estimated brain age by our method, which is again 80 years old, but estimated by our method, we can see that their brains look more or less similar, despite that their real chronological age can be very different, right? So this means our estimated brain age is indeed a better marker for confined health condition, the health condition of the brain. Okay, so some further analysis here. In this experiment, we did not only look at the brain age itself, but we also look at the slope, which is the changing speed of brain over visits. And we can see here that for the control cohort, you have a changing speed approximately equal to 1, which basically means, well, if you grow older by a year, your brain also gets older by a year. But if you now look at the Alzheimer's cohort, their brain aging speed it's much larger than 1, which again indicates that this disease accelerates neural loss. And as expected, this MCI cohort, mild cognitive impairment cohort, falls in the middle, because we know that MCI actually represents the intermediate stage between being healthy and Alzheimer's disease. And again, if we stratify this left plot with respect to the chronological age of the subjects, we can see that again, the most accelerated aging effect happens for those young Alzheimer's patients. And to further illustrate the strengths of the method, we actually applied it to a second data set, which includes healthy controls and patients with alcohol use disorder. So for this alcohol data set, age range is much wider, because you remember for ADNI participants, it's beyond 60 years old, but for the alcohol data set, it basically covers the whole adulthood from early 20s to late 80s. And then now if we look at the estimated brain age for all the healthy control participants, you can kind of see a nonlinear developmental trajectory from 20 to 80 years old. And if we again overlay the brain age, the estimated brain age of the alcohol patients and patients with alcohol use disorder, on top of the healthy trajectory, it turns out that the effect of alcohol use disorder is more pronounced at older ages. And this is different from the ADNI result. If you remember, the disease effect is more severe at younger ages. And the reason for this pattern for the alcohol data set is that alcohol use disorder is more of a chronological disease in the sense that the disease effect accumulates over time with chronologic drinking. And lastly, we were able to show that AUD also causes accelerated aging effect, because the changing speed of brain age between visits, across visits, is significantly larger than one, and than that of the healthy control cohort. Okay, and remember that our framework is an autoencoder. So that means, given the latent representation, I can reconstruct a brain out of it based on the decoder network. In other words, we're able to generate synthetic brains of different brain ages by traversing the latent representation along the disentangled direction tau in the latent space. And we did exactly that for the two data sets, where we simulated typical brains of different brain ages. And we can see that the developmental pattern is most similar for the overlapping age range between the two data sets. But we also noticed that for the alcohol data set, the aging effect is less, it's more subtle compared to the effect in the ADNI data set. And this is because we have very few samples near age 85. So probably the method becomes quite conservative in extrapolating the age effect for the alcohol data set. Okay, so far all the results are presented are based on this longitudinal self-supervised disentanglement. And it turns out this approach can also be used as a pre-training strategy to provide a better image representations for other downstream tasks. So here I used a pre-trained latent representation to perform classification between diagnosis label. Notably for acne data set, we predict whether a subject is healthy control or Alzheimer's disease, and for the alcohol data set, we predict healthy versus alcohol use disorder. And the upshot is that in most settings, the representations pre-trained by our method LSSL provide the most accurate classification results. I guess I can go into the results in this table in more details if anybody is interested in this later. But I think the general take-home message is that our longitudinal self-supervision can be considered as a very compelling pre-training strategy for performing downstream tasks using longitudinal data. And lastly, this is another piece of evidence. So if you plot the training curves, accuracy curves for all the experimental settings in the last slide, it is still mostly the case that our method results in faster convergence because our pre-trained latent representations provide a better initialization for the downstream model. Okay, so in the end, I do want to re-emphasize the strength of our longitudinal self-supervision. In fact, the concept of using pairwise data to exploit within subject dependency has stimulated multiple directions in our lab, mostly led by me or our student, Jiahong Ouyang. And here I'll briefly mention 3 follow-up works that leverage this concept of longitudinal self-supervision to improve representation learning. I guess I'll just give an overarching idea of each work, but won't go into detailed results of these works due to the time constraint. Okay, so the first improvement in this paper, and this is actually done by our student Jiahong Ouyang, and this paper was actually the Young Scientist Award runners-up in the MICAI conference in 2021. So the one observation she had was that, well, in the original LSSL formulation, the aging direction is confined to be a linear direction. This is a really harsh constraint, considering the space of MRI data can be highly nonlinear, and many people have shown that fact. And her idea is that instead of confining to be a linear direction, how about we just let it be a smooth trajectory field instead of a linear trajectory field. So this trajectory field, as long as it's smooth, it can be either linear or nonlinear. And so she proposed this longitudinal neighborhood embedding method, which basically says that for any trajectory vector connecting 2 visits of the subject, I will look at its neighborhood in the latent space, and I'll then encourage that all the vectors that fall into this neighborhood all should progress along a similar direction. So this is the concept of neighborhood smoothness. And by imposing that regularization, if you will, she was able to capture or get this smooth trajectory field capturing the brain aging pattern in the latent space. And you do want to note that if you use a regular autoencoder, this nice smooth vector field cannot be reviewed in the latent space. And moreover, the latent representations pre-trained by this method actually outperform the ones pre-trained by LSSL and also all the other baselines that I showed you earlier. And this result indicates that by injecting this nonlinearity into the model, you may have a better flexibility and capacity for training downstream tasks. And actually, the extension of this work was invited to submit to the special issue of the Journal of Medical Image Analysis, and is currently under review. And next, we also extend this longitudinal self-supervised setup into a multimodal setting. So the idea here is that instead of learning the aging pattern in just one imaging modality, you can actually find coupled developmental effects across multiple modalities. So the setting here is that for each subject, you have a pair of visits at 2 time points. And at each time point, we also have 2 neuroimaging modalities. For example, at each visit, we can get an anatomical T1 weighted MRI for describing the morphology of the brain. And simultaneously, you can get a different imaging modality, for example, diffusion weighted MRI, to quantify the wet matter microstructure of the brain. So basically, each training sample or each training bundle, should be the case in this setting, is composed of 4 images, essentially 2 time points times 2 modalities. And for each modality, you can basically learn this latent space. And in either latent space, we can estimate disentangle a latent direction separately in each space, such that the developmental effects in each modality, when projected onto this direction, can maximally correlate between each other, between the modalities. So this concept is very similar to the traditional canonical correlation analysis, if you remember, which basically aims to find the most coupled information between 2 modalities of data. Here we just extended to the longitudinal setting, where the goal is to find the most coupled developmental effects between the 2 modalities, because apparently this correlation is performed on these delta vectors or developmental vectors between visits. So I guess I'm not gonna go into details of this detailed results of the paper, but anyone interested in this can find the MICAC paper and the source code online. And lastly, we also tried to further disentangle a second direction associated with disease progression, in addition to the aging direction that I just mentioned in the original LSSL work. And the idea is that here we assume that the control group and the disease group can share the same distribution in their normal aging speed, normal aging pattern, but only the diseased subjects can have a progression in terms of the disease status. So only along this disease, the second disentangled direction, you would observe a significant group difference between controls and diseased, but there's no group difference in terms of normal aging between the 2 cohorts. And it turns out that with this regularization, we have a chance to disentangle the disease effect in addition to the aging effect. But what we did here a little bit differently is that we ended up having to propose this self-supervised idea. Essentially, it means that if the diseased cohort has multiple subtypes, for example, in the case of cognitive impairment, you have either stable MCI, progressive MCI, and Alzheimer's disease. So you have those 3 subtypes in the disease cohort. During training, I only provide a binary label of each subject, being either a healthy control or a disease subject, but without providing the actual subtype label. And it turns out that having this weak label can even further help disentangling the disease direction, and the latent representations can result in much better downstream task classification of the subtypes, for example, classifying stable versus progressive MCI. So I know these last few slides can be really head-waving, but I just hope that these slides can become some sort of teaser that can convince you that the specific type of pairwise training, longitudinal pairwise training we adopt here is really a powerful approach in leveraging the strengths of the repeated measure design. To better learn image representations of the data, and this idea has been continuously motivating some of the ongoing research in the lab, and I'm quite excited to see where this longitudinal self-supervision can eventually lead us to. So I guess I'll stop here, and we can go to some of the questions we have.
Wonderful. Thank you so much for a great presentation. I really enjoyed, and it's very well aligned and related to some of our applications. There are a couple of questions already typed in, and the floor is also open for everyone in the audience to start typing in your questions in the Q&A box, or if you want, if you prefer, you can raise your hands and we can unmute you. So maybe we can start by a question asked by one of our PhD students, Caroline. She says, it seems like, sorry, SimClear is a close competitor of this longitudinal method. And SimClear includes true contrastive learning, sorry, with attractive and repulsive forces. Are there any practical advantages or disadvantages of SimClear versus LSSL? And can you envision a way to incorporate contrastive forces, negative examples into LSSL? Perhaps the final project you mentioned goes in that direction.
Yes, so this is a really good question, and I guess I can answer that from different aspects. First of all, result-wise, just confined to this LSSL work, and we also saw that we compare our LSSL with SimClear, and turns out that at least for these applications, this longitudinal self-supervision can achieve better downstream classification results. And the major difference you already mentioned in your question is that SimClear is more towards the, the common perception of self-supervision, which involves constructing this positive training samples and negative training samples, the so-called concept of contrastive learning, and use these positive pairs and negative pairs to jointly train the model. And here, LSSL, I guess you can think of it as a method that only involves positive pairs. So you can think of the, the within subject pairs within each subject trajectory of 2 visits. You can think of that pair as a positive pair because apparently the 2 data points belong to the same subject, and you want to encourage some sort of dependency or some specific longitudinal relationship between that pair of MRI points. So that can be regarded as a positive pair. And it's a really good idea to also incorporate the negative pairs into this framework. I guess one of the extensions done in this work by Jia Hong, which is actually a further extension of this Nikai paper, which is being under review in media, incorporates this negative pairs concept, where I believe she incorporates this prototype training, where she could cluster these longitudinal trajectories to incorporate both within subject relationship and also between subject relationship. So that's the direction we're trying to converging towards in general the concept of SimClear. I hope that answers the question.
Thank you. There's another question in the chat box, and that's how do you learn the direction of the tau vector? And I guess that question is, you want to minimize the angle between different movements along the edge, right, and make them parallel to tau, but how that direction of tau could be arbitrary. How do you fix that or learn that?
Right, so that's a good question. So the trick here is that you want to learn this autoencoder model. You want to learn the latent representations of the data, but simultaneously you also want to estimate this direction. So the direction is parameterized by a vector in your training. So if you have your latent space is, I don't know, 1024, then you have a vector of 1024, a unit vector, to be estimated by our method. So essentially, this is joint estimated in your framework. It's estimated in a way that this direction unit vector should basically coincide with all those subject-specific within subject progression vectors. And that's the general goal of this framework, which is also represented in this objective function where you optimize your encoder and decoder G and D, but you also want to optimize the vector, the unit vector, the direct one direction tau, such that this tau can be parallel with all those pairs of MRI data. So I guess that answers this.
Thank you. There's another question in the Q&A box. This one from a postdoctoral fellow, Kite. Leaf is asking whether there are any methods within the ones that you've developed that you could employ to identify regions of the brain that change the most, for example, temporal lobes in Alzheimer's disease.
Yes, so that should be a major direction if we want to deploy this method to actually clinical usage, to advance discovery of a certain neurological disease. And one way to do that is, I would assume it's based on this brain synthesis by traversing along either your brain age direction or disease direction, and you reconstruct the brain associated with different ages or associated with different disease types, and to see their difference across different disease subtypes. But I want to note that at least in the work of this LSSL, this original work, one disadvantage is the use of autoencoder. We know that autoencoder cannot reconstruct very sharp output of MRI data, especially if the data is in 3D. Then what you observe is that you have a lot of these smooth out reconstructions, which might limit your capability in finding these specific differences in local brain regions. And one direction we're working on to address this issue, to replace the autoencoder with this more recent normalized flow model, which guarantees more to some extent to be able to reconstruct a sharper image data with fine-grained morphological structure, so you can actually have a better understanding which brain structure, local brain structure, can be affected also in a much more clear fashion.
Great, thank you. I have a question myself. So the tau vector that is learned is a direction in the latent space.
Right.
How do you convert the units of length in that direction to a year? How much of that you consider to be 1 year?
Oh, that's a really good question. So that's one specific I didn't mention. So basically, when you estimate all these, we did a group level normalization by essentially matching the mean and standard deviation of the estimations to the real chronological age in years. But you are totally right. The estimate.
So once you have that direct vector, you use the healthy controls.
Yes.
To find the origin, to fix the origin of that vector, like the 0 years old.
Right.
And also the unit length of that, the length that corresponds to 1 year in the healthy.
Yes. Yes.
Okay, so I guess that also answers my next questions, which was why is it that in slide 20 and 24 and 25, the average for controls is 1 year in the horizontal, in the chronological age and projection age. It's like, yeah, exactly on that one. Just slide 24. Yeah, this one, the blue bar, for the control, the average in the chronological age is exactly 1. And also,
yeah,
that's because you fixed it to be 1.
By construction, yes,
Yeah,
to some extent, I agree with that. It has a certain effect to have this normalization effect. But again, this slope is computed within each subject, whereas the normalization, it's computed over the group level. But I totally get your point. I think you have a valid concern here. I guess the more important information in this figure is that it's the relative difference between the 3 cohorts that makes sense.
Yes, absolutely. Yes,
right.
Very good. Thank you. I see there's another question in Q&A from Caroline again. A bit off topic, but I saw on your website some more work on interpretability, specifically training deep models to interpret deep models, which seemed train again like model To convert MRIs from one class to another in a cycle-consistent fashion, can you briefly summarize the key contributions of this work and next steps you're excited about in interpretability for medical applications?
Yes, so if I understand the question correctly, yes. So there's one work in last year, where we I was trying to remember that work, where we designed this framework that can simulate, given a healthy brain, we can simulate what the brain looks like if the person contracted the disease. And inversely, if we give a diseased brain, I can simulate what if the person is cured, what the healthy brain of that person would look like. And by doing that, you can basically compare the simulated healthy and diseased brains to infer what's the disease effect. So that's on the interpretability of the model. So that's the work we did there. So I think for that work, it aligns with a major research project nowadays, which is a counterfactual learning. So the counterfactual learning generally refers to making tweaks in your learned latent space such that you can simulate your own data if one factor was changed to a certain extent. And that is generally believed to be a better interpretability method compared to traditional interpretability method based on computing saliency maps from your learned model. And the extension of that counterfactual simulation we're working on right now is to use that concept in this causal modeling in terms of analyzing the effect of confounders on your neuroimaging data. So I don't know if anybody knows this concept of confounders, but it's extraneous variables that influence your input MRI and outcome disease label, and it can just mess up your model training. And we need to understand how each confounding variable can influence your MRI brain appearance exactly. And the way to do that, we're currently thinking, is to incorporate that counterfactual model to be able to simulate your brain, for example, what your brain looks like if you transfer from male to female or you grow older barrier. So that's the direction we're trying to work on, not from the interpretability perspective, but from the causal modeling of confounders. So that's so stay tuned. We're working on the topic right now.
Wonderful, thank you.
Yes.
I don't see any other questions in the Q&A box. If anyone has a question, you can type it now. Otherwise, it's past 3 o'clock. We're already past our time by a couple of minutes. So we can thank our speakers for a wonderful presentation and also for answering our questions.
Yeah, definitely. Thank you for having me here again.
Yes.
Always communicate more.
This was wonderful and very much related and relevant to our work. So greatly appreciate it. And I see there's a couple of chat messages also thanking you.
Thank you. Okay.
So everyone in the audience, please join me in a virtual round of applause.
Okay. I guess I'll see you later.
Bye.
Bye-bye.